everybody. Welcome to our recorded lecture entitled Pacemaker Cell Physiology. As we talked about in class, as we were covering Chapter 19, the heart, I told you that I was going to be covering basically the portion regarding pacemaker cell physiology on connect integrity, and then the rest of Chapter 19, which will pretty much be the cardiac cycle and cardiac output, we'll cover in class. So, let's start with trying to find my slide here that I want to start with, so give me a second. I'm trying to locate it. Let's see. Okay. Let's start. I'll start right here, this slide. So if you have your notes, you can also start, or I should say also grab those and get to this slide, which says anatomic structures controlling heart activity. So I'll give you a second. Now, when you look at figure 1914A, you see the cells in yellow here, and these are called pacemaker cells. And these pacemaker cells set the pace of the heart. They really have a very minimal contractile activity, almost none. And so their goal is to tell the contractile cells to contract. And so, most of us would say if we, tested our resting heart rate, via maybe a radial pulse, we'd find that our heart beats 70 times per minute. Well, what sets that pace at rest, and what allows the heart rate to speed up or slow down? And these are these pacemaker cells. Now, if you anatomically identify them, as you will in lab, you'll see that the master pacemaker, the SA node, is located in the upper right atrium. And then there's the AV node kind of located in the lower right atrium. And then that AV node splits into what's called the bundle of His, or the right, and you can see it's indicated here, the atrioventricular bundle, which also is called the bundle of HIS, bundle of His, and then that splits into, in the septal area, two right and left bundle branches, which further splits into Purkinje fibers, which you can see here in terms of that. So kind of the pathway, oops, I'm sorry, I did not mean to change slides. Let me go back here, sorry about that. The pathway for electrical, sorry, I'm having some technical difficulties here. The pathway for electrical conduction would be SA node sends its electrical impulses to the AV node, which sends its electrical impulses to the atrioventricular bundle, or the bundle of His, which you can see I'm tracing it now. And then the bundle of His breaks up into right and left bundle branches, which is in the septal area. And then the right and left bundle branches further break into what are called Purkinje fibers, which line the ventricles, which you can see here. And actually, the flow of the pacemaker cell electricity is the same flow that would occur as blood is flowing from atria to ventricles, right? Right side, left side, I'm doing now. And then from ventricles to either the aorta on the left side or to the pulmonary trunk on the right side, which is right here. You can see it here, which then would go to the lungs, right? So that's kind of the anatomy portion of what you're looking at. So now, we say, okay, let's think a little bit more about, well, okay, so does anything control these pacemaker cells? Well, yes, but as we'll find out shortly, pacemaker cells can set their own pace, but we do have some controlling features of the pacemaker cells, and that would come from our autonomic nervous system, which is part of our central nervous system, and you guys talked about the autonomic nervous system in Bio 144. There are two arms of the autonomic nervous system. Remember, the autonomic nervous system we don't have control over, and so we have a sympathetic and parasympathetic division within the autonomic nervous system. And we remember the sympathetic division, as you can see up here, was our fight or flight division. So if we were in an emergency, or if we write this here and then if we were anxious, exercising, scared, the sympathetic nervous system would be directed to predominate and would cause the classic responses of increased heart rate, increased blood pressure, pupil dilation, preparing us for an emergency, basically. Now, in terms of the autonomic nervous system, we have a cardiac center that's found in our that's found in our spinal cord area, or our brain stem, I should say, our brain stem, and that's called the cardiovascular control center. And there are two kind of sub-portions of the cardiovascular control center. There's the acceleratory center, which is the sympathetic nervous system. In terms of, as you're looking at it here, you're seeing if you look over here, the sympathetic color here, especially that which would be innervating the SA node, which I'll circle, and the AV node, which is circle, and the sympathetic nervous system can also innervate your myocardial tissue. So it has, from this cardiovascular control center, which you can see here in terms of coming down. Remember, the sympathetic nervous system has, those are the parasympathetic nerves. The sympathetic nervous system here's in green, you can see, has the ability to innervate, several different areas in the heart. And we say it's part of the cardiovascular acceleratory center. And there's two neurons here you can see. First neuron, second neuron. First synapse, second synapse. You can see that in terms of what you're looking for. We're trying to speed up not only the heart rate via the SA node and the AV node, but we're also trying to speed up the force of contraction of the ventricles, which we'll eventually talk about later in class. And so if the sympathetic nervous system predominates, well, and if we speed up these two pacemaker cells, then obviously my heart rate's going to increase, right? And now we think about, well, what happens if the parasympathetic nervous system controls or is predominating? Well, then let's erase all this. Then, oops, get a different color. Then we have thinking about these are antagonistic systems. So when the parasympathetic system predominates, as you learned back in Bio 144, rest, digest, and restorative things happen. Things that we would think about as resting, things about restoring, going back to normal, and our digestion is controlled by the parasympathetic nervous system, which obviously we're not talking about now, but rest, digest, and restore. So when the parasympathetic system predominates, which you can see the parasympathetic system is part of that cardiovascular control center in your brainstem, particularly medulla, you have a cardioinhibitory center, and that's what the parasympathetic system where we're talking about is involved. And the parasympathetic system, you can see, has you can see the neuronal wires, and they're in yellow, or I guess a goldish color, and I'm tracing them right now so you can kind of see that. And now, the key thing with this parasympathetic system, and I'm going to circle once again, it only can control the SA node and the AV node. It doesn't control the myocardial tissue. So you don't see any gold wiring, parasympathetic wiring, getting down to the myocardial tissue here. You don't see that. And so when the parasympathetic system predominates, it only can control heart rate, and heart rate drops. And that's the opposite effect of what the sympathetic system does, and that's why they're called antagonistic systems. When you need your heart rate to speed up, your sympathetic system will predominate, and the cardio-acceleratory center will send messages to the SA node, AV node to increase heart rate, as well as to increase the force of contraction of the ventricles, the myocardial tissue, to contract with more force. If the parasympathetic system predominates, then you get messages coming on the parasympathetic neurons to the SA node and AV node, which tell them to slow down and slow down heart rate. Now here's the biggest thing to keep in mind here. The autonomic nervous system, with its two divisions, only can modify cardiac activity. It's not telling the heart, okay, start beating. The heart can beat on its own, as we'll find out, and so this autonomic nervous system will actually modify cardiac activity. It can influence heart rate as well as force of contractions with the sympathetic nervous system, and it can influence heart rate with the parasympathetic nervous system. Now, you do have sensory neurons, which, if I grab a different color here, you can see here in blue, and I'm making a red arrow so you can see that red arrows point to the blue, that we do have sensory neurons that can sense when I should speed up heart rate, when I should slow it down, or when I should, make the sympathetic nervous system be in charge, or when the parasympathetic nervous system is in charge. And those, which you can see in blue, are considered sensory input. We get baroreceptors from our aortic arch, as well as baroreceptors in my carotid artery. And baroreceptors can detect blood pressure, BP. And so if you think about, what if these baroreceptors detect that blood pressure is below normal? Well, then you want to think about a negative feedback that the body needs to make sure quickly that we get our blood pressure back up to normal. We'll actually do a blood pressure lab here pretty soon. And that's negative feedback. So, very, very simply, I'll abbreviate. To do this, if you're thinking about which system should predominate, it should be the sympathetic system. Because in order to get my blood pressure back up to normal, I need to get my heart rate to speed up. And as we'll see, the force of contraction of the heart can also speed up, and that can lead to increased BP. And then vice versa, well, what if the opposite occurred? What if my heart rate was high? Maybe I just exercised and I want to restore it or return it back to normal. Again, you can see the opposite change for negative feedback. So I've got above normal blood pressure. Maybe I just exercised, but I want it to return it to normal. Then that's when the parasympathetic system will predominate, and it can control heart rate. Oops, so heart rate will drop. Heart rate will drop, and that will help drop blood pressure back to normal. Okay, so there's a quick little thing on how the autonomic nervous system works, and let's move on to the next slide here. Here's a different look at autonomic nervous system innervation of the heart. I like this one because it's very concise. You can see the sympathetic nervous system here, innervating the SA node, the AV node, and the ventral myocardium. And you can see the parasympathetic system only innervating the SA node and the AV node, just kind of what we talked about before. Now, thinking about all this, the heart contraction itself, the heart's ability to move blood throughout the heart and eventually out of the heart, involves two events. You've got to have a conduction system, that's your pacemaker cells, that will tell the heart how many times to beat per minute. The cells of the conduction system are called pacemaker cells, and you also may hear them called nodal cells because you hear the word SA node, AV node, that kind of thing. And as I just mentioned before, the SA node is the master. It starts the whole process of this, how many times the heart should beat per minute. Then eventually those pacemaker cells have to somehow tell the cardiac muscle cells that actually contract to contract. And they're both going to involve electricity called action potentials, which we'll look at. The cardiac muscle cells that contract, this process first happens in the atria and then spreads to the ventricles, which that's how we want blood to flow, right? So that makes sense. So, let's move along. Now, there's going to be a term that I'll write up here. Now, I've got half the term here, but I'm going to write the full term when we think about the heart itself, heart contraction. We say a heart can contract because it undergoes something called excitation, contraction, coupling. Put this. And it's a fancy phrase for basically saying in order for the heart to contract, it has to be excited to contract. The excitement part comes from the pacemaker cells, okay? And you can see here that the SA node at the top of the right atrium actually initiates pacemaker electricity called action potentials, spreads it to the left atrium, and then spreads that impulse to the AV node, which eventually will spread down the bundle of His, down the bundle branches, and then the Purkinje fibers. And so we're basically trying to tell the heart how many times to beat per minute. So the whole process is called excitation, contraction, coupling. What you're seeing right now is just the excitation part. So let me get rid of my phrase here. Right? Okay, there you go, there you go. And let's move on. Now the contraction part. So when you think about contraction, you guys talked about skeletal muscle physiology a little bit back in Bio 144, and you talked about sarcomeres and how in a skeletal muscle cell, in order for muscle contractions to occur, thin and thick filaments have to slide past one another, sarcomeres shorten. When sarcomeres shorten, myofibrils shorten. When myofibrils shorten, the muscle cell shortens, which you see here. When the muscle cell shortens in a skeletal muscle, the whole muscle cell will contract. Well, in cardiac muscle, we kind of look at the same thing, only cardiac muscles are branched. They have these special intercalated discs that we talked about in lab, and these discs are going to help us to spread this electricity throughout all these cardiac muscle cells, in essence, to eventually allow them to contract so that we can get blood flow from atria to ventricles and eventually from ventricles to the rest of the body or the lungs. So, first, we're going to look at the pacemaker cells and what their action potential electricity looks like. And we're going to think about, in order for all this electricity to occur, and we'll go back to this picture. The SA node has to start the process. It's the master pacemaker. And we'll see what happens if it weren't to work properly here in a second. So let's go back here. All right. Now, these cells in the SA node, we say they spontaneously depolarize, or they're autorhythmic. They don't really need an outside influence to be able to initiate a heartbeat. Now, we just talked about the autonomic nervous system that could modify heart rate. Well, if this were to happen, if you were to take out of a live animal, if you were to take a heart and remove the heart, and I'll draw my version of a heart, we'll put the chambers there, atria, ventricles, and you were to remove all sympathetic and all parasympathetic wiring, so no autonomic nervous system stimulation. You put this heart in a special tray, and you gave it what it needs, oxygen, electrolytes, etc. This heart would continue to beat on its own, autorhythmically, without any outside influence until its solution ran out. And so that tells you that the heart's autorhythmic. While the autonomic nervous system can control or can modify heart rate, truly the heart is autorhythmic. And the SA node starts at autorhythmicity, and all nodal cells are considered autorhythmic. But because the SA node's the master pacemaker, its firing rate at rest, how many times it's going to tell the AV node, the bundle of His, the Purkinje fibers, etc., how many times to beat per minute, is based on the set rate of the SA node. So the SA node is going to say, okay, all nodal cells at rest are going to beat 70 to 80 times per minute. And that means the AV node will know that, the bundle of His, the bundle branches, the Purkinje fibers. So, well, what would happen? Oops, there I go again. Sorry. Let me back up. I can't see. Give me a second here. Sorry about that. Getting a little ahead of myself. What would happen if something were to happen to the SA node? So we'll make that apparent by going, okay, we don't have an SA node anymore. Maybe it stopped working. Well, then you're left with the AV node being able to spontaneously depolarize. But notice, look what happens. It can only tell the rest of those pacemaker cells or nodal cells to only beat at 40 to 60 times per minute, which we could survive, but we'd have a little trouble. Now, what if we lost the AV node then? Well, then the bundle of His, it does have an inherent ability, autorhythmic ability to beat, but it can only tell the rest of the heart to beat 20 to 40 times per minute, or the Purkinje fibers, and this wouldn't be good enough. So if someone has their master pacemaker, their SA node, that doesn't work, they have what's called a pacemaker surgery, and they will replace the SA node with some special wires. And the wires, if I can get to it, if it lets me, let me go backwards here. Sorry, things are going real slow on my computer right now, and I'm not sure why. Hopefully we don't have a freezing moment. But if the SA node has to be replaced, what we really do is come from the outside of the body, and we put special wires to it, and we control it with an external pacemaker, which maybe some of you may know someone that has that. All right, so we're going to move on here. Now, figure 1916 actually shows the electricity that you would see in an SA node. You also see with the same electricity in an AV node, in a bundle His, in a bundle branch, and also Purkinje cells. But because the SA node is the master pacemaker, we look at that first. All right? Now, the thing I want you to focus on is the fact that this action potential is showing the electricity that's involved in an SA node. It's autorhythmic, spontaneous depolarization. So you can see, I want to point this out. Let me go into my mode so you can see a pen. Want to point this out. that is almost looks like a ramp. It's also called a pacemaker potential, and it's a fancy name for the fact that a SA node really doesn't have what we would call a perfect, steady resting membrane potential. Now, it's indicated that the resting membrane potential is at negative 60 millivolts, but it actually varies between negative 60 and negative 40. And you can see the threshold here, which is negative 40, in a SA nodal cell or in any nodal cell. This threshold is automatically reached. So the resting membrane potential kind of varies. It spontaneously depolarizes. Now, you talked about depolarization as you get more positively charged, moving further away from the resting membrane potential, in this case, which is anywhere between 60 and negative 40 or negative 41, I guess. Spontaneously, this SA node will reach threshold on its own. We don't need any outside influence to do that. And we're able to do that because there are special sodium channels that will spontaneously open, as you can see here, and allow sodium to actually move in. And this arrow is wrong in terms of, oh, okay, no, it's not. We're showing, this is the cytosol. I'm sorry. The cytosol is here, and the extracellular fluid is here. So sodium will move from where it's highest to lowest in. And as that's happening, these special sodium channels can then start to also open some calcium channels, and that eventually helps me to reach threshold. And when I get to threshold, then I get into phase 2. Now, there's other special calcium channels that aren't shown here. This calcium channel is a little bit different. It's called a fast voltage gated calcium channel. So once this is happening spontaneously, remember, these sodium channels spontaneously open, sodium influx occurs, moves into the cell. There's some special calcium channels that aren't shown here, but that will also cause me to get depolarization spontaneously without any outside influence, no neurotransmitters, no hormones. Then that triggers depolarization. In purple here, I've circled it, which is phase 2. Now, depolarization in phase 2 involves, now you're thinking about, maybe you thought back to your nervous system chapters in Bio 144, and you thought about the action potential in a neuron, and you're like, the action potential in a neuron had sodium influx for depolarization. Well, in a heart SA node, it's calcium influx. So calcium moves in and causes us to further depolarize, get even more positively charged inside this SA nodal cell. That eventually leads to repolarization in yellow here, and you can see repolarization moving down. And that allows now potassium to exit. Special potassium channels open and potassium exits. So to reach threshold really quickly, in an SA nodal cell, we have something called a pacemaker potential, which is kind of a resting membrane potential that varies between negative 60 and negative 70. It automatically will reach the threshold voltage with the help of special sodium channels opening and sodium influx occurring, and also some calcium channels that aren't shown here. And then we'll reach threshold, and we'll get a depolarization phase here in number 2, which will get fast calcium channels to open and calcium influx to occur, and that will depolarize me. You can see my numbers here. And then repolarization occurs, in which my potassium channels will open, potassium efflux will occur, and I'll get to somewhere around that pacemaker potential again, and I'll start the whole process over again. So if you imagine at rest, your heart rate's at 70 beats per minute, you'd see 70 of these action potentials, these electrical changes. Now, this is what we visualize, obviously, mathematically in a graph, but in reality in the cell, you wouldn't see these bell-shaped curves. You would see just changes in membrane potential with changes in different ions. Positive ions moving in to get more positive, or positive ions moving out to get more negative, which you talked, returning to the pacemaker potential, which you talked about ions and those types of things back in Bio 144. All right, so now let's move along. What I did for you is took this figure and then gave you a bigger picture of it, so you could see now what actually causes. And so this is the pacemaker potential. We reach threshold. This is depolarization. This is repolarization, and what ions are involved, which you should know whether they're moving in or out, or etc. And you should also be able to look at this and recognize that the shape of it is a pacemaker potential, because you're going to see something different with the contractile cells next. Now, this type of electricity, these action potentials, the ion movement is going to spread, as we talked about, from the atria, right and left, to the AV node, to the bundle of His, AV bundle of His, to the bundle branches, to the Purkinje fibers. And then that will, as we'll see, will help us to eventually allow blood to flow from atria to ventricles to out of the ventricles. Now, so eventually we got to get cells to contract. It's great that we can have pacemaker cells that set the pace for the heart, but we need cells to actually contract and eject blood. Notice the difference in a cardiac cell that actually has the ability to contract. So cardiac cells have the ability to contract. Notice the difference in the action potential. Let's just take a peek again. There's the cardiac cell that actually can contract action potential, the electricity that allows it to contract. And once again, what we just looked at, there is a pacemaker cell. Now, this is not electricity, that's going to go, okay, SA node contract. The SA node doesn't contract. The SA node is setting the pace for the heart. This electricity will eventually be sent to other nodal cells, and eventually as we'll see, nodal cells can be connected to contractile cells, and eventually a contractile cell will get this message to contract, which you can see here. This is the electrical message. And when you look at a contractile cell around the sarcolemma, you can see different voltage-gated channels. And now this contractile cell has a steady resting membrane potential of negative 90 millivolts. It waits to be told time to contract. And so once you see that, we were told through a gap junction to contract, then we'll move into what's called depolarization. And depolarization, you'll see some special sodium voltage-gated channels opening and sodium influx occurring very quickly. That will make that resting membrane potential become more positive. That's what depolarization means. We'll get a bunch of positive charges moving in to the cardiac muscle cell. And then we do something interesting. We have what's called a plateau phase. We don't repolarize straight down. We have a plateau, and the plateau in purple is really important here. The plateau starts out by special voltage-gated potassium channels opening, but they open pretty slowly. And so we get a little bit of potassium exit, which can drop me, positive charges leaving, can make me become a little bit, what we say, less positive. So, plus 29, plus 28, but we don't drop straight down back to the resting membrane potential, because there are special slow voltage-gated calcium channels that open during this plateau, and calcium influx occurs. And that keeps me kind of leveled off, so I don't drop quickly to the resting membrane potential. Finally, there will be those calcium channels will close, and most of the potassium voltage-gated channels will open, and potassium will exit the cell, exit the cardiac muscle cell, so efflux. And those positive charges that leave rather quickly start to drop me down. Positive charges leaving, negative 10, negative 20, negative 30, and I get back to the resting membrane potential. Now, if you looked at this up close, this is what it would look like. You can see the sodium voltage-gated channels, influx occurring. I didn't put the potassium channels here because the main event of this plateau is calcium influx through special calcium channels. And then repolarization is potassium efflux or exit. Efflux means exit. Now, let's put this all together. Remember, I used the term excitation contraction coupling, which you can see here. Now, I want to orient you. This is not a page out of your book. This is from a different textbook. But what it shows nicely is this could be a pacemaker cell, autorhythmic cell, and this could be a contractile cell. And so how did the pacemaker talk to the contractile cell? It talks through a gap junction. And these are found, remember, in intercalated discs, right? That you will have or have looked at in lab. As part of the intercalated disc that we're focusing on will be the gap junction. Now, there's no chemical synapses in cardiac muscle, like with a neurotransmitter and stuff, like there was in skeletal muscle. there is what we call electrical synapses. And these electrical synapses occur because ions will move from one pacemaker cell to a contractile cell, or from a contractile cell to a contractile cell. So when you see those intercalated discs, inside there's a gap junction and allows ions to move through, and that's how one cell can stimulate the next. You don't have neurotransmitters that are in a synapse, a chemical synapse. You have ions that have been released, either moved into a cell or as you can see, and then we'll eventually move into the next cell in terms of that. And so if you're a contractile cell, that will cause you to contract. So you can see the ion movement here will eventually lead to this contractile cell going through its contractile cell action potential, and eventually you can see I have the plateau phase here. Now what's not shown here is the sodium influx, the depolarizing phase, the repolarizing phase. But remember, we need to get the cell excited. We need it to reach its depolarizing current, and so that's how the one contractile cell can tell the next one contract. And when you imagine the heart, atria contracting to get blood to the ventricles, and the ventricles contracting to get blood either to the lungs or to the rest of the body, the cells have to be told to do that. Now, you talked about in Bio 144 about skeletal muscle, actin and myosin. Here's some actin molecules. Here's some myosin heads. You talked about the sarcoplasmic reticulum being the storage depot for calcium, and you talked about T-tubules being, the invaginations of the sarcolemma. And allowing the electricity to get down deep into the myofibrils and cause sliding filament model to occur, so that myosin made contact with actin and shortened sarcomeres. Now, I can't go through all that again. You can obviously go back and look at your Bio 144 notes, but what I want you to realize is something pretty amazing happens in your cardiac muscle, contractile cells, and it's called CICR. C here stands for calcium. You can write that down here. I stands for induced. C stands for calcium. And R stands for release. Let me go through that again. The first C here stands for calcium. Write that down. The I stands for induced. Write that down. The C stands for calcium. Write it down, and the R stands for release. So calcium induced, calcium release. What's interesting about cardiac muscle is once the depolarizing current comes and we get the depolarization, we have a plateau phase. And here's where the plateau phase where calcium influx occurs. And if you don't recall that, I'll go backward in a second. We'll look at the action potential again. You can remember that. That calcium influx leads to calcium efflux or release. Calcium induced, calcium release. And that buys us time. It gives us a little bit of time, this plateau electricity, to actually physically fill the ventricles with blood. Because that's the main event, the ventricles contracting. And so instead of repolarizing straight down, we've got this depolarization, we got this little plateau, and then we repolarize. Instead of going straight down, which wouldn't give my ventricles time to fill, we have this plateau phase, which gives my ventricles time to fill electrically. And that's what the calcium-induced calcium release is all about. Now, once the calcium comes out of the sarcoplasmic reticulum, it binds to the actin molecule, specifically troponin, which I'm not going to ask you on a test because you've already been tested on that. But that allows myosin heads to bind, sarcomeres to shorten, and this contractile cell to contract. And what ends up happening is we've excited the contractile cell via another contractile cell or a pacemaker cell, and that leads to contraction. The coupling part is putting these two things together. Let's move on here. Now, what's interesting, and it'd be nice if we could have time to talk about skeletal muscle again, but we just don't. You've already covered that in Bio 144. So, you've talked about tetanus in terms of complete contraction of skeletal muscle and no relaxation. Well, cardiac muscle can't have complete contraction. It has to have a really long refractory period, meaning the heart can contract and relax before it can be stimulated again. And any electrical cells can't fire a new impulse during a refractory period. So your heart can't just have this sustained contraction without relaxing. If we didn't relax, I mean, that would be a bad thing. We probably wouldn't live as long as we'd want to. There I go again. Darn it. Sorry. Let me go back. Do this. We would, our heart wouldn't last as long as we'd want it to, but really, remember I said the plateau phase really contributes to this refractory period. And the heart can contract and relax, and when the heart relaxes, it gives the heart time to fill properly, which is really important. Okay. Boom. All right, so now this next slide under EKG, we're actually going to physically cover this in our next lab, in the EKG lab. I won't be asking you questions on our test on this. I think I might have accidentally put it into our review sheet. I won't be asking us test questions on the ECG because we're actually going to cover it in our next lab, and there will be a lab quiz based on that. So we'll cover it that way. So to be continued to talk about the electrocardiogram. To be continued. To be continued. All right. So I'm going to stop there. And what we've covered basically was talking about how the autonomic nerve. Well, first we introduced you to the pacemaker cells, where they were located anatomically. We then talked about the autonomic nervous system and how it could modify cardiac activity like heart rate and contractile activity, but doesn't initiate it. We then talked about the two types of cells, pacemaker cells and cardiac muscle cells. We talked about the term excitation, contraction, coupling, and then really physically looked at what a SA node action potential or a nodal cell action potential would look like. You should be able to recognize that and also think about the different ions that are moving in or out during the different phases of the pacemaker potential, depolarization and repolarization. And then we compared that with the cardiac contractile cell, which looks a lot different, which you should be able to recognize. And again, the phases of depolarization, the plateau phase, repolarization, and the ions that create that. So that's all for this recording. Certainly, if you have any questions, please visit me during office hours. Have a good day.